Good evening, friends. This is Dr. Kabir Sardana from Delhi, and my talk is on repurposing drugs for COVID-19. We have been using various drugs in the last 2 years, and over the years we found that some of these drugs were not really working. So, this is the resident pocket notes of our department when our PGs go and work in COVID wards. And this is as early as April 2021. This is what they used to carry with them. This is what even we used to have with us. So there was this protocol of giving certain drugs in the first 7 days of the disease. So doxycycline was there, ivermectin was there, you had paracetamol, which probably made sense, and multiple vitamins. And then, of course, when the patient went from mild to moderate disease, the drugs changed to methylprednisolone, enoxaparin, which actually has saved lives, and Clexane. And of course, when they went to a severe form of disease, like moderate to severe disease as per protocol, then of course everything was there. You had to give steroids, dexamethasone, Clexane, ecosprin, statins, metformin, remdesivir, tocilizumab, plasma, and of course antibiotics. Now, of course, a lot of these things are not there in every patient, but this was the protocol being followed. And of course, this protocol is different from what was there a year back when we had hardly any evidence for any of these medicines, especially prednisolone. So, what we must understand is that therapy has gone changed over the years, and the therapy of COVID-19 depends a lot on the stage. This is a diagram to show you the stages of COVID-19. So you have the incubation period, wherein of course no one comes to us. Then you have the symptomatic phase, day 5 to day 11, wherein you have an RT-PCR positivity, you have viral replication that tends to then go down. And then of course you have the one that is the problematic stage, where you have immune response, cytokine release, T-cell dysfunction, and of course sometimes, unfortunately, death. So the importance of knowing this is that all the drugs that have been tried in COVID-19, whether they've been tried in India or abroad or in research laboratories, target either the first step or the second step. So that is the symptom that you see during each step. As time goes by, the lung infiltration goes up and the symptoms become more severe. And so hence, you can have drugs working on the virus, that's the antiviral drugs at the first phase, or the drugs working on the inflammation. So I have divided them into two categories, two simple categories to make you understand, because some of the drugs that we give, like remdesivir, has got no role beyond day 11. So we should know when to give what. So that's how you can divide the different drugs for COVID-19. Now, if you look at the drug discovery, now, there are three ways of discovering a drug for COVID-19. The first, of course, is a direct COVID-19 specific antiviral drug, and this is not feasible in epidemics. It takes years to do this research, and frankly speaking, there is not a single anti-COVID-19 specific drug as of today. The second option, you screen existing databases. This is called the in silico approach. So they have computer simulation models where you can find out numerous drugs that work in a particular disorder. Not necessarily always true. And the third one is using pre-existing components, or what is called existing antiviral drugs. So there are drugs that have been used for COVID-19 are actually used in Ebola, Zika virus, and other anti-RNA viruses. Some of them are even DNA virus drugs being tried. So one must understand that what we are using today, or maybe a year from now, are not exactly COVID-19 specific drugs. Okay, right. So now come to the variables. There are so many variables in drug discovery. The first is in silico. In silico is a very fancy thing. You can publish articles on in silico, effect of drugs, but then does it really translate into clinical efficacy? In silico means you use computer simulations to think of an ideal drug targeting the components of the SARS-CoV-2. Then comes to in vitro, which is actually what we should do, the step 2. We should always do in vitro research. This is very relevant for COVID-19 and also for dermatophytosis, wherein we are giving fancy drugs to our patients without having any in vitro data. It is really comical. For example, voriconazole in dermatophytosis is totally absurd, and hence you should always look at in vitro data. Third, of course, is in vivo simulation, in vitro, in vivo giving to patients in live. Then the biggest problem, there are three variables to decide drug efficacy in COVID-19: the severity of disease, the stage of disease, and of course the last, the golden thing, RCT. And RCTs, very few RCTs exist, and hence we have very few high-level quality drugs for COVID-19. Nevertheless, so if you look at a diagrammatic visual of how the virus affects, the first step, of course, is the virus enters the target host cell, the lung epithelial cell, via its receptors. So there are two receptors. One is the ACE2 receptor, and second is the TMR-SS2 receptor. You see in the diagram. Then the virus replicates, translates, matures, and at that point of time, there is a release of pyrins that cause amount of fever, bit of malaise and myalgias. That is stage one. The stage two is actually very crucial. And then the virus actually goes inside the alveolar macrophages and epithelial cells and stimulates the first host immune defense mechanisms, wherein it actually simulates certain interleukins, IP-10, and with the interferon gamma feedback mechanism, stimulates T cells, monocytes, and macrophages. Now, this is going to happen to almost every patient. Now, what happens next is what is important. If things are in a bad shape, the red arrow, you have a cytokine response, production of cytokines, IL-6, IP-10, interferon gamma, IL-2, 10, and you have non-neutralizing antibodies that do not really help. They may cause more damage, and the patient goes into a cytokine storm. And if, of course, the patient has got an intrinsically good immunity, which is the case in a majority of patients, the alveolar macrophages clear up the virus, neutralizing antibodies inactivate the virus, the macrophages destroy the virus, and then the CD4 T cells and the CD8 T cells eliminate the virus. So this is what is the good and the bad one. So of course, my discussion is not going to be on the last slide in this particular panel, but the first two scenarios. So let's look at the first two scenarios. So the antiviral drugs. So the antiviral drugs work on the first stage. Now you can have drugs which actually block the first stage, that is the stage wherein the virus confabulates with the receptors, that is the SARS-CoV-2 or the other receptors. So you have some very fancy new molecule drugs in this category, which are actually the hot drugs, which actually can stop this particular virus from actually entering into the epithelial cell. Then you have, of course, the virus enters the cell through endocytosis. Nothing is there for that. Then there's a fusion of virus into endosomes leading to genomic release. So in this you had HCQS. You've got an antibiotic called ticlocanin and of course Viracept. Then you've got drugs working on the replication of genome. A lot of drugs in this category, including remdesivir, ribavirin, interferons, emitin, of course arimitin. And then the next stage is translation of virus proteins. Again, you have a host of drugs in that particular category, some of which I will quickly summarize for you. And then, of course, the overview. No? So these are the antiviral drugs, starting from receptor blockage drugs to other categories. Now, in this particular drug, the first one is hot. It is going to be probably the most important drug in the future if you can make in vivo translation. Viracept again is the same. Some of them are out of the picture. And so if you look at these categories, this is what's left. These are the four classes of drugs that actually have got potential in COVID-19 stage 1. So I'm going to discuss a few of these drugs and talk about what are these drugs, where are they available, what is their efficacy, and what is their status. So let's come to the first drug, eloxystatin. This is a very interesting molecule which actually blocks cellular entry of SARS-CoV-2 remarkably by blocking something called cathepsin L. So it's a drug that blocks at the first step. So the virus never enters the cell. It's under research. It's under active research in Italy, Europe. It's an antiviral drug primarily under research, not available in India, but this is one of those categories that actually is going to be hot in the future. Second drug, of course, alloredonis. These two drugs, nifamostat and chemostat, actually the word stat means stop. They actually block the entry by blocking a very important receptor called the TMPRSS2. Very long sounding name, but this is a remarkably effective drug and it prevents coagulation. It's very interesting to very diverse actions. There's a dosimetry described. There are trials that have been done both in vivo, in vitro, and you never know, this may come maybe by the end of the year, not available in our country, unluckily. Then you have Viracept. Viracept is a very interesting molecule because it's available in our country, incidentally. It is actually used for HIV. It's a membrane fusion inhibitor, again blocks the COV2 spike protein. Our dose is not exactly established as yet. It's being tried in certain parts of China and Italy and even Europe, and even some trials are on in UK. The important thing about this drug is it's there in our country. So, if this works, we'll have a novel repurposed antiviral drug for COVID-19. Remdesivir, which is the hot new, which is a drug which is written by everybody. It's basically antiviral DNA polymerase. It has got 2 regimens, a 5-day, 10-day regimen. There are multiple studies. The double-blind RCTs have found to be useful in certain categories of patients, not in all patients, not to be given in mild moderate disease. The exact duration is also controversial, but ideal duration is first 10 days where the virus proliferates. It has got side effects. There are patients who died of side effects, but it is the only antiviral drug that has been given FDA approval, whatever be the merits of the approval. Now, the question that you must ask, which I always ask, is that is it really superior to other anti-COVID virus drugs? There is no head-on-head trial of this drug with any of the antiviral drugs in COVID-19. So one doesn't really know if it is so supremely effective, but it's got some evidence for its use, but it should be used in the first 10 days only. Then, of course, you've got this molecule. This is a very interesting molecule. It's a mAb. It's an Ig1 monoclonal antibody against a receptor binding domain of SARS-CoV-2 protein. It is given as a single dose IV. It's a remarkably effective antiviral drug, but strictly to be given in the first 10 days, within the first 10 days of symptom onset or a positive RT-PCR. This drug is useful and important because in November 2020, the US FDA gave this approval for use in non-hospitalized adults. That makes it an interesting drug because FDA approvals don't come so easily. The drawback, of course, you've got to catch a patient in the first 10 days, which is not going to be simple because the first 10 days include, patient coming to you, going to some Hakim. So that's the problem. But if you catch them in the first 10 days, this would work very well because it will block the drug from entering the host macrophage. Then, of course, emetine. Emetine is a very simple drug. It actually is an anti-emetic drug. It's remarkably cheap, available in our country. It works against antiviral DNA polymerase, same as remdesivir. It's been tried in vitro, and it has been tried previously in Zika virus and Ebola virus. And as an extension here, the interesting part of the drug is twofold. One is its EC50 is very low. That is, the effective concentration for efficacy is low. And number 2, it synergizes with remdesivir. So that's the very interesting part. 2 drugs that can synergize at the same site, one cheap, one effective. Maybe you could use remdesivir and then probably go on to emetine as an oral drug for anti-SARS-CoV-2. Then, of course, this is a drug that's been used and abused. Again, antiviral DNA polymerase. The problem with this drug is the remarkably high dose. It's like popping in so many pills. It has been known to cause decreased viral load, which it would, because it's an antiviral drug. And of course, some patients have improvement, but only in mild to moderate cases. And of course, approved initially by the Chinese. That's how it caught on in the world. And my query always is that, this efficacy of the drug is very uncertain compared to other antiviral drugs. And most importantly, can somebody take so many pills? Look at the dosimetry. It is amazingly high dosimetry. People have to pop in this pill and plus everything else. So I do not know whether in actual in vivo scenarios, this would really help or not. But it is very much there. Ivermectin. Now, of course, ivermectin is a drug of dermatologists, and this drug, we see WhatsApp discussion, and people say, where is the proof? What is the evidence of ivermectin and all that? And we have great ability of, disregarding our own work, surprisingly. Ivermectin has got a lot of evidence. In fact, there are studies on almost all viruses, RNA viruses, Zika virus has been tried in, Anopheles mosquitoes also incidentally. It has got a very discrete mode of action. It inhibits the IMP alpha beta, which is actually a receptor that helps to block the entry of most viruses into the body, including SARS-CoV-2. The problem with this drug is twofold. One is, of course, the evidence is there. There are multiple studies in vitro and in vivo. Many studies published on PubMed, unlike the preprints. The problem is the dose. The dose is variable. In an active infection, it may range from 12 mg, 24 mg, BD for 5 days. It is only useful in the first 7 days, not otherwise. Advantage is very easily available. And I think that what probably is missed out is that, if you combine this with doxycycline, the results actually are superior. Now come to data on this drug, because it's one of my, favorite drugs actually. And I have been taking it for like 5 months now. So I know it actually helps. The evidence on this is huge. There are controlled trials, there are RCT trials, there are prophylactic trials. And I always find it very amusing when people say, where is the proof? Where is the proof? I really wonder why people don't go to the PubMed and start searching for ivermectin studies. There is a lot of proof. Anyways, now come to another interesting drug, niclosamide. Niclosamide again is an anti-helminthic drug. Inhibits antiviral translation. It's an antiviral drug. It is known in vitro to cause a rapid decrease in viral load. In one small study, it is known to also help in clinical improvement. The important thing now, come to the important part. With such a little evidence, niclosamide, you will not find many papers. It has been given phase 2 trial license in India. Why it's been given phase 2 license in India, we all know. I mean, some of us know. It is backed by one of the most powerful, industrial house in the country. And the question that I always want to keep asking is, why is this drug being rubbished and ivermectin? Why is this drug being trialed and ivermectin rubbish? They both are the same category. The studies for ivermectin are many more than niclosamide. So remember that a lot of drugs that have been trialed and then something is rubbish. We have to rubbish one drug to trial another drug. So I think we should take a relook at antihelminths, including ivermectin. Niclosamide, of course, is in phase 2, and I'm sure it'll probably come out by the end of the year. Come to the second part. The second part is the inflammatory phase. Okay, the one that's a problem. So when the patient virus enters the macrophage and the host cannot hold it back, then you have the inflammatory phase. In that, you have a lot of cytokines being released. You've got a cytokine response, and of course, the patient gets into trouble. So here is the next category of drugs. The first is corticosteroids. I will not dwell on this. It's well established. It works on every cytokine. Then you've got IL-6 inhibitors, tocilizumab and sarilumab. You've got the TNF-alpha inhibitors now, thalidomide is being tried. Then anakinra was tried for a short while. Then you've got these fancy molecules, bradykinin inhibitors, or even colchicine, which is a cheap molecule inhibiting the neutrophil response. And then, of course, your Indian hot new drug, the 2-deoxy-D-glucose, which actually works on cytokine storm. Doesn't really work as an antiviral drug. So, which of these drugs or more are useful and will be useful? Well, the first, thalidomide. Thalidomide is a drug that we use a lot. Dermatologists use a lot. It inhibits TNF-alpha primarily. That is the perceived action in case of COVID-19, dose 100 mg per day. Trials are underway, and I think this might be something very useful if proved to be superior to other drugs. This is a very interesting drug. I didn't mention it previously, bevacizumab. This is a well-established drug for anti-cancer therapy, inhibits VEGF. And VEGF inhibitors are important because when the VEGF level goes up, there is hypoxia, inflammation, and severe inflammation in the inflammatory tract, which is what is seen in COVID-19. It is given with 0.9% saline over 7 days. There are trials underway. It's a really remarkable molecule as an adjuvant, and it's under trial and it's available in India. So, this is a molecule that you can watch for. It might really help a lot. Tocilizumab inhibits interleukin 6. Doses vary. It is inflammatory. It's very favorable. Studies have been seen in CT findings and CRP level and fever counts and lymphocyte counts. It is used by a lot of us in severe cases, but I'll give you a take on this in the end as to what is the catch in tocilizumab, available in our country, of course. Now come to the catch. You see, all these drugs have been used. They sound very fancy, but there are two important things to understand. Number one, there is never any head-to-head trial with steroids. Never. And corticosteroids inhibit every cytokine. So no company does it. You can ask them why. And of course, the question is why. Well, they don't do it because, if you compare corticosteroids with, which is, mind you, has passed a double-blind RCT, you might find that nothing is as good as corticosteroids. So I think that all these new molecule drugs, except for the VEG inhibitor, probably are overstated in their capability of use. The kind of steroids we give in a country probably inhibit all cytokines. Okay. So what has happened in the last 2 years is that we've made mistakes and we have learned what is not great, like doxycycline is not great, isotretinoin is not great, vitamin C is not great. So I think there's nothing wrong in making mistakes, because mistakes is a method of improving yourself. It's as Billy Joe Armstrong said, it's better than doing nothing. So what's left of this whole story? Well, if you look at the phases of the COVID-19 symptom complex, you will realize that there are just two great classes of drugs. Antiviral drugs, there are lots of drugs. I mean, I don't know what I can put my finger on. I would put a finger on Admeton if I had a choice. And unless and until you can go and prove an in vivo study, and then do an S2S study, head-to-head study, one cannot say that one drug is superior to the other drug. You know, I think remdesivir, even today, I believe, is a highly awaited drug. But let's see what happens. Of course, in the anti-inflammatory spectrum, steroids are established: tocilizumab, serlimumab, colchicine, thalidomide, and of course heparin. I don't think that much will happen here because what we have are extremely good drugs. I don't think you will have anything new coming out in this particular segment of drugs. So that's like how it is. Of course, I'll come to the last, second last slide. That is the principal trial group. You must log in once in 3 weeks. This is a fantastic trial group. University of Oxford. It is on the NHS panel, and the principal trial group actually is a platform of randomized trials or treatments in various disorders of pandemic importance. And I'll give you a take on what they have done on the last 6 months and why they're important to know. These are the guys who recently have now investigated a trial on Ayurvedic, which we keep on rubbishing in India. You know, we have armchair experts who say a Bhagavas drug hai, without even reading on it, so they are back on it. And so it's important to not disregard your own evidence. They are the people who began a trial on colchicine just in March 2021. They are the guys who said that probably favipiravir could be used in patients at home recovery, and these are the guys who found that the two antibiotics that were raised last year are rubbish. So the evidence on COVID-19 is evolving. It is our job to simulate, understand the facts, and differentiate between preprint data, PubMed data. Okay, if you do that, you probably understand things better. Of course, I'll end my slide with my favorite quote. As this famous saying from this famous movie, Shah Rukh Khan said, "Picture abhi baki hai mere dost." So I guess there's a lot more which may happen in the next 1 year, and maybe I'm proved wrong. But I personally believe that the spectrum of newer drugs repurposing is probably in antiviral drugs, not in the anti-inflammatory drugs. Thank you very much, and I hope you have a great day.